new method, a step towards future 3D printing of human tissues, a study by University of Sydney. A team of bioengineers and biomedical scientists from the University of Sydney and the Children's Medical Research Institute, CMRI, at Westmead have used 3D photolithographic printing to create a complex environment for assembling tissue that mimics the architecture of a human organ. The teams were led by Professor Hala Zregat and Dr. Peter Newman of the University of Sydney School of Biomedical Engineering, and developmental biologist Professor Patrick Tam, who leads the CMRI's embryology research unit, and the paper was published in Advanced Sciences. Using bioengineering and cell culture methods, the technique was used to instruct stem cells derived from blood cells or skin cells to become specialized cells that can assemble into an organ-like structure, similar to how the needle of a record player navigates the vinyl grooves to create music. Cells use strategically positioned proteins and mechanical triggers to navigate through their intricate environment, replicating developmental processes. The team's latest research employed microscopic mechanical and chemical signals to recreate the cellular activities during development. Professor Hala Rekat said, "Our new method serves as an instructional manual for the cells, allowing them to create tissues that are better organized and more closely resemble their natural counterparts. This is an important step towards being able to 3D print out working tissues and organs," he said. Dr. Newman said building tissues from cells requires detailed instruction, not dissimilar to constructing a building from many different parts. Imagine trying to build a Lego castle by randomly scattering the blocks on a table, hoping they'll fall into the correct place. Even though each block is designed to connect with others, without a clear plan, you'd likely end up with something that looks more like a large pile of disconnected Lego blocks rather than a castle. The same can be said about building organs and tissues from cells. Without specific instructions, the cells would likely group together unpredictably within the incorrect structures. What we've effectively done is create a step-by-step process that guides each building block to exactly where it should go and how it should connect with the others, Dr. Newman said. In line with this approach, our recently published work applies a new 3D printing method to define instructions for cells that guide them into forming more organized and accurate structures. And through this, we've created a bone-fat assembly that resembles the structure of bone and an assembly of tissue that resembles process during early mammalian development. Research into complex tissue and organ-like structures known as organoids help researchers understand how organs develop and function and how diseases affect the organ may be caused by genetic mutations and developmental errors. The knowledge gleaned from the study also enables the development of cell and gene therapy for diseases. The ability to generate the desired cell types further provides the capacity to produce clinical relevant stem cells for therapeutic purposes. Professor Hala Zrekat said, beyond understanding the intricate instructional manual of life, this method has immense practical implications. For instance, in regenerative medicine, where there is a pressing need for organ transplants, further research using this approach may facilitate the growth of functioning tissue in the lab. Imagine a future where the wait list for organ transplants could be drastically reduced because we can generate such tissues in the lab that sufficiently resemble their natural counterparts. Dr. Newman said, moreover, this technology could revolutionize how we study and understand diseases. By creating accurate models of diseased tissue, we can observe disease progression and treatment responses in a controlled environment. We hope this could one day lead to more effective treatments and even cures for diseases that are currently hard to tackle. Professor Tam from CMR said, in the past, stem cells were grown to generate many cell types, but we could not control how they differentiate and assemble in 3D. With this bioengineering technology, we can now direct the stem cells to form specific cell types and organize these cells properly in time and space, thereby recapitulating the real-life development of an organ. The researchers are hopeful that the research will have the potential for treating vision loss caused by conditions such as macular degeneration or inherited diseases causing loss of retinal photoreceptor cells. Professor Tam said if we can generate a patch of cells by bioengineering and see how the whole system functions, then we can investigate therapies that use functional cells to replace cells in the eye that were lost because of disease. It would have great impact if we can deliver healthy cells into the eye. Regardless of whether the macula, the area of the retina responsible for cellular vision, had been lost due to inherited disease or because of trauma, the treatment could be the same. The idea of treating rare genetic diseases and improving quality of life in this way is empowering. We expect that this work will lead to advanced therapies that can be moved into practice. The team will next focus on furthering the technique to advance the field of regenerative medicine and potentially new treatment approaches for many diseases. And this is from University of Sydney on phys.org. Please leave your comments and thank you for your support. Please support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.